Hello, everyone. Thank you so much for joining us. We are extremely excited to have you with us for this live kickoff event. My name is Joshua Kroll. I'm the Director of Sales and Marketing with Avexia Diagnostics, and I will be your host for today's webinar. I want to welcome you all to this very special webinar regarding the power of genetics and the introduction and overview of the 3x4 test. Today, I have the distinct pleasure of being joined by an outstanding guest, Dr. Yael Joffe. She will be our presenter for the webinar. For those of you who don't know, Dr. Joffe, in her role as Chief Science Officer, Dr. Joffe brings over 20 years of experience in nutrition, genetics, sound research, clinical practice, and scientific integrity to 3x4. Now, in the rapidly evolving discipline of nutrigenomics, Yael is globally acknowledged as a leading expert in the field. She is highly sought after as a speaker, has co-authored 2 books, It's Not Just Your Genes and Genes to Plate, and has been published in multiple peer-reviewed scientific journals. Yael is at the forefront of nutrigenomic education and has helped develop and supervise nutrigenomic courses around the world, including her own Manuka courses, which have trained hundreds of healthcare practitioners globally. And if that wasn't enough, she is also currently an adjunct professor teaching nutrigenomics at Rutgers University and Maryland University of Integrative Health. Now, before we get started, I would ask that you please type any questions you may have into the chat box on the right-hand side of your screen in that little toolbar, and we will do our best to address them at the end of the presentation. Now, keep in mind, if we don't get to everyone's questions during the webinar, I will be following up with everyone individually afterwards. Please note also that we have included a sample report for your reference, which I think is going to be very helpful as Dr. Joffe goes through her presentation. This can be accessed through the handout section in the toolbar, again, on the right-hand side. Now, without further ado, Dr. Yael Joffe.
Thanks, Joshua. Thank you for the introduction, and apologies for the delay. We were trying to work out how to get my camera working so that I was hoping you could actually see me speaking and not just my slides. But nonetheless, the most important thing is the content. So we will be introduced to each other visually at another time. And for now, let's jump in and get going. So about, what are we now? Middle of 2021. So about a year and a half ago, I took a team of colleagues to the American College of Nutrition conference in San Diego when we were still attending in-person conferences. And what we wanted to get a sense of the practitioners at the conference was, how are you using genetics in your practice? What is your sense of it? What is happening for you? And I want to just share with you a couple of results with regard that we glean from these practitioners, remembering that the practitioners that attend ACN are what we would call our kind of advanced, already converted, like preaching to the converted practitioners, right? So what did we find of the 100 practitioners we interviewed? We found that 61% did not have the content knowledge to confidently work with genetic tests. 52% did not know how to translate the genetic test into practice. 47% felt there was no supporting community for them as health practitioners. And most alarming of all for me is this one, that 74% of these nutritionists attending ACN did not know which genetic test to trust. Now, for me, who has been in the nutrigenomic industry for more than 20 years now, this was a complete fail. That after 20 years of work, practitioners still did not have enough knowledge, they did not know where to go for knowledge, and most importantly, they did not trust what was happening in the industry or know how to ask questions of a company to determine what company they should trust. So this got me thinking of why had we failed? Why had we not 18 years, 19 years into this industry not had a different outcome? And I want to share just a little bit of my thinking with you. So the first thing is I was thinking about the science. So even though the Human Genome Project was launched in 2003, and we were all extremely excited about the potential of having our full genome available to us, what I believe has happened in the marketplace is that we have actually overstretched the science. That in our enthusiasm to build commercial genetic tests, we have made the science do more than it is actually capable of. And in doing so, we have lost the trust of both the consumer and of you, the practitioner. The second thing I was thinking about is, what is the experience of our customers, of your patients, in how they engage with genetics? Whether it's 23andMe or whether it's a practitioner-based genetic test, our general sense is that there was a sense of underwhelming, that they were promised that genetics would offer them this hyper-personalization, the ability to really create a personalized diet and lifestyle for themselves. But when they received their reports, they didn't find that it was particularly different from what they were experiencing around them. And lastly, you, the practitioner. So you can see we've got a bit of a grumpy practitioner here. And the reason being is when we looked back over the 19 years, we realized that in truth, the undergraduate curriculum hadn't changed significantly, no matter what your undergraduate degree. So nutrigenomics and genetics was not being taught in our medical degrees in such a way as to make it clinically useful to us. We didn't have a support group or a community. We didn't know how to ask tough questions and trust that we would get a credible and true answer. And for many practitioners, it was more a burden of time and of learning and of seeking out education than anything. That if you have to spend 3-4 hours trying to interpret a genetic test before you see your patient, that for you as a practitioner is a burden. That is not adding value to either you as a practitioner or to your patients or to your practice. I really believe that we are better than this. And I really believe that we can do genetics a whole lot better than this. So it started thinking about what are the possible solutions? We've identified the problems, but now how can we solve them? And a lot of these ideas around finding solutions was really why I founded 3x4 Genetics. It was having this disillusionment and this disappointment in all I was seeing around me in the industry for such a long time, and feeling that I really could build a company that could try and fix things. So I want to share with you today, depending where you are, what are some of the things that we built to try and address some of these problems in the market. There's 3 things that we're essentially trying to do. The first is we're trying to provide actionable and personalized insights for your patient through you, the practitioner. We want to ensure it's practitioner guided. One of the problems of a direct-to-consumer without a practitioner is we land up dumbing down the science, dumbing down the value and the gems in genetics, in which case we land up selling data instead of selling clinical value. And the third thing is it needs to be rooted in science. And we're going to talk about that quite a lot. There needs to be a strong foundation in science that you can trust. We wanted to be a genetic partner for practitioners who what we call are visionary practitioners, who see the future, who know that no matter which way they turn, genetics will be part of your future. Whether you choose to start testing today, tomorrow, or next week, you need to understand that genetics is part of the healthcare market, of the ecosystem. And we as a company wanted to make sure that we could bring practitioners into a place where we could resolve some of the issues that I addressed. So what makes 3x4 different from all these other companies that I've been referring to, that you have engaged with up until now, that potentially you have worked with or you have read about, or that your patients have brought you reports and said, can you please explain this to me? So for me, it wasn't just about building a better report. It wasn't just about adding another 100 genes in and saying, I've got more genes than your test has got. The problems we had could not be fixed by just fixing the report. We had to fix the industry. We had to fix everything that was happening around the nutrigenomic space. And that meant addressing the report, dealing with the education of nutrigenomics, speaking about mentorship, and we'll talk about the difference between education and mentorship, and finally, building a community. So I'm going to go into detail on each of these, but wanting to understand that in order to change the way genetic testing is done in the marketplace, you need to be able to address and build all four of these pillars. So let's start with my favorite, which is the report. So we knew that in the report, there were two key issues we needed to address. The first is, could we build a genetic report that was science that you could trust? So we could change that 74% did not know which genetic test to trust to knowing exactly which genetic test to trust. The second part, which I will deal with afterwards, is how we engage with these genetic reports, how our patients engage, and I'll deal with that in the next one. But let's start by talking about how do we build science we can trust? Now, one of the major problems in the industry is this concept of the single SNP. So I don't know if all of you who are listening know about the SNP. It's a single nucleotide polymorphism. It's sometimes referred to as a polymorphism or a gene variant, but it's basically a spelling change in our DNA that gives us information about something that may have changed in our physiology. Now, up until recently, the entire industry has built nutrigenomic tests on the concept of the single SNP. And what I mean by that is if you look at any report, it'll say, here's, I'm going to use MTHFR. You're all familiar with MTHFR, I'm sure. Here's MTHFR. And because you have this gene variant or the SNP, or I've appallingly seen things like mutation and defect, you will need to take this supplement and do this diet and do this lifestyle. And I call this SNP-based treatment. Now, it was only 5 or 6 years ago when I realized for myself that the way I had been building genetic tests for the first decade and a half had been fundamentally wrong. That I had been part of this movement which had built a genetic test saying, if you have this SNP, take this supplement, do this diet. If you have this SNP, etc., etc. And what I realized when I really looked at the science is that these single SNPs are not that powerful. Remember we spoke about overreaching the science? That a single SNP by itself does not diagnose disease, it does not cause disease, and by itself, it cannot be the answer to a diet plan or to a single supplement. So if the single SNP as a foundation of building genetics is essentially incorrect, what was the correct answer? So I started looking at what else was going on in the literature when I came across some beautiful literature around polygenic risk scores. So the concept of polygenic risk scores is we don't just take one single SNP, we take multiple SNPs. So let's use the example of diabetes. So what they did in the research is they took a group of diabetics and a group of non-diabetics as a control, and they identified all the gene variants that potentially played a role in diabetes, and they did a genetic test, and then for every allele, so when you have one copy from your mom, one copy from your dad, so an individual for a particular SNP will either have one copy or two copies, they would add them up and they would come up with a score. So the more gene variants you had, the higher the score you had. And what they were trying to understand is, if I have a higher score, am I more likely to have diabetes? And if I have a low score, am I more likely to be in the control group? And this is what they found, that the higher scores were associated with the condition, whether it's diabetes or cardiovascular disease, obesity, arthritis, and the lower scores meant a lower likelihood of having these conditions. And these scores are called polygenic risk scores. Polygenic because it's multiple genes that come together rather than a single SNP. Now, what I realized is all this amazing validated research had been done around disease conditions, diabetes, heart disease, obesity, etc. But we know, those of us who love to be in part of the functional and integrative medicine, that we don't want to define our patients by the disease that have been labeled with. We want to understand what happened upstream that may have caused some physiological dysfunction that would manifest as diabetes downstream. So if that was the case, then surely the answer was to build polygenic risk scores upstream. So what I did was come up with this idea of pathway-based analysis, where we look at biochemical pathways, and I'll take you through some of these, biochemical pathways that are further upstream, more root cause analysis, and build polygenic risk scores around the biochemical pathways rather than building polygenic risk scores around a disease condition. And that is what we have done at 3x4. We do not offer recommendations on single SNPs, but we do offer recommendations around these polygenic pathways. So we took a validated methodology, but applied it to an upstream medicine. This is an amazing research paper that came out, I think it was in August last year, by Fahed. And why I love it so much is up until recently, we had this idea that there was a world apart between medical genetics and nutrigenomics, which is the space we are in. Medical genetics is when we look at gene variants, so they're also SNPs, but they're quite impactful, they're quite powerful, or the correct word in genetics is penetrant. So the higher the penetrance, the more likely an individual is going to have that condition if they have the gene variant. Low penetrance means that by itself, that gene variant is not going to cause a disease, but if we group them together, we can increase their power, which is what I was referring to in the slides before. But what is so exciting about this paper is what they showed, that these monogenic variants, which are these high impact medical ones, an example would be BRCA, the BRCA gene, BRCA1, associated with a high incidence of breast cancer, FH, familial hypercholesterolemia gene, associated with very, very high lipid levels, that these were regarded as monogenic high penetrance. But when you look at the BRCA gene, just because you have you do a genetic test and you're positive for the BRCA gene does not mean you are going to get breast cancer. And in fact, it's anywhere between 40% and 80%. Now that's a huge difference. If I'm positive for the breast cancer gene, that's BRCA, and you tell me it could be 40% or it could be 80%, that's a really big difference to me. And what this paper found is that the explanation of why you would be 40% versus 80%, most of that explanation resided in what was happening in the polygenic background, the polygenic environment. So when we spoke about polygenic risk scores, these low penetrance ones, the nutrigenomic ones, that aren't very powerful by themselves, but when they come together, they have an impact. This almost like a genetic ecosystem would determine whether these medical genetic like BRCA would express themselves and manifest in a disease condition like breast cancer. So for the first time, we found a way that made sense to us of the relationship between these genes that can have a higher impact on disease and the kind of testing that we do at 3x4 Genetics. The other thing that's so important to know is that not all SNPs are made equal. And I know this might seem obvious, but what I'm trying to get across here is the idea that there are SNPs and SNPs. And when I spoke about impact, so I don't like to talk about risk because we're not diagnostic, we're not medical. We're talking about how do gene variants impact our physiology, but some gene variants will have a greater impact than others. So when we talk about SNPs, for those of you that are familiar with them, like MTHFR and ApoE and COMT, they are quite impactful. When we talk about other ones like LPL or CATP, they have a lower impact. So how do we determine, how do we understand how impactful these SNPs are? So we needed something at 3x4 to be able to independently build a way to tell you how impactful that SNP is, to tell you and your patient. And up until then, it was done mostly by reading the literature and having an opinion. But we wanted a methodology that was so transparent and so independent that if I didn't do it, any one of the scientists in my team could build a genetic test using the methodology. And so I'd like to introduce you to Darwin. We called it Darwin because the concept of Darwin is only the fittest genes survive. Whatever gene variants, whatever SNPs we put into our 3x4 genetic test, we wanted to make sure that only the best made it into the report. And so let me tell you about Darwin. So Darwin was based on the concept of scientific validity and clinical utility. Scientific validity means the science is great, it's good enough. I've read all the articles, I've checked all the original studies, and we'll talk, and I am happy. They were good journals, the stats were good, the sample size was big enough, they were well validated, but in some way, the science was good enough, and I get a big tick. Now, most genetic testing companies in the marketplace will stop at scientific validity, and for many years, so did I. If I saw enough scientific research around a SNP, I was comfortable to be able to put into a genetic test. But what I realized, what my great epiphany was, is that that's not enough. It's not enough just to be able to say, if the science is good enough, it's good enough to put into your test. And the reason being that you are using these tests with patients. If it's not clinically useful to you, it does not matter if it's scientifically valid. There has to be clinical value to you and your patient to justify your patient spending money on this test. If it's only scientific validity, we are selling your patient data. But what we really want to offer is clinical usefulness. So we built Darwin to have 8 criteria. 4 criteria that make a scientific validity, and 4 criteria for clinical utility. Now, I'm not going to go through all of them, but I'm going to give you some insight into some of those criteria, and you'll get a sense of the kind of scoring. And for each of these criteria, every single SNP is scored. So the first one is biological plausibility. So this asks the question, if I'm looking at the COMT gene variant and the pathway is estrogen metabolism, is it biologically plausible that this gene variant plays a role in this pathway? And I think we can all appreciate that in the case of COMT, it does impact estrogen metabolism. But of course, that's not good enough. We need to document every single research paper. We need to look at the stats. Was it in vivo? Was it in vitro? Was it replicated? But was it biologically plausible is very important. Does it make biological sense? The other thing is the gene variant, is the SNP specific to the pathway? So let's talk about COMT again. So when we look at the biochemistry, we do a lot of biochemistry, right? Because when we look at the biochemistry, what does the COMT SNP impact? It impacts methylation, it impacts estrogen metabolism, it impacts mood in terms of dopamine and serotonin. Does it impact, for example, adipogenesis, the ability to break down adipose tissue? Well, we couldn't find any research. So we actually do Darwin on every SNP in every pathway that's relevant. So it's not just that the SNP has to have good science and it has to be clinically useful, it has to have good science that relates to the pathway we're testing and clinically useful to the pathway we're testing. And this is often where the SNPs land up falling down. Another one is bioimpact. Can we show that if you have this gene variant, it's going to change the biochemistry in some way? So what we're not interested, so when nutrigenomics started, it was like, This SNP is associated with cardiovascular disease. Those are called association studies. But for me, that's too far apart. It's not good enough to say TNF alpha is associated with cardiovascular disease. I want to know, what did TNF alpha change in the biochemistry that in some way would impact cardiovascular? Did it change CRP? Did it change TNF alpha? Did it set off an inflammatory cytokine storm? What was the biochemical impact? So we have based our scoring on biochemistry, not on association studies. This is part of our clinical utility. If I test for something and I get an answer, is there something I can do? Because if I'm just giving you an answer, but there's nothing you can change with your patient, there's nothing your patient can do differently, is that clinically useful to your patient? Absolutely not. So is there an intervention is very important for scoring. This is actually the final criteria, and it's my favorite. And the idea behind this is, if I do this genetic test with my patient, will it provide me with clinical insights that will help me make a different and better decision for my patient than if I didn't do the genetic test? So does it give me something more than dietary guidelines could have given me without my patient spending money on their test? Is there a value differential? So this is the purest example of clinical utility, of clinical usefulness. Does it help me make a better and different decision? Right. I'm going to give you two examples of genes to illustrate how we apply the scientific validity and clinical utility. So the first gene is a very famous one. It's called GSTM1, which is glutathione S-transferase M1. And the reason we've got a garbage truck behind is because the GST gene is very, 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 very well known for its work in phase 2 detoxification. So the job of GST is to take an activated toxin molecule from phase 1. In phase 1, toxin molecules, whether endogenous or exogenous, get activated. They're at their most dangerous. They add a glutathione molecule on, which makes them water soluble. And once that water soluble, it can be excreted for the body. So GSTM1 is an incredibly important enzyme, obviously made by the gene, to be able to clear toxins from the body. Now, there is a gene variant which results in a deletion of this gene, something that we test for in a genetic test, that if you have the gene variant, you will not have this GSTM1 functional, which means your ability to clear toxins in phase 2 will be impacted, not gone, impacted. The reason it's not gone is there are many genes that work in phase 2, and we want to understand, using a polygenic risk score, are there other genes or is it just GM1? Like, how impacted is my phase 2? So the question is this a scientifically useful gene? Is the science good enough? Absolutely. I've been studying this gene for probably more than 20 years. It's got hundreds and hundreds, if not thousands, of articles that show that the GSTM1 gene, when it has a gene variant, impacts the biochemistry by deleting the enzyme. And it has been shown when the enzyme is deleted, there is a decrease in the clearance of activated toxins in phase 2. So on all levels, we've shown biochemical impact, biological plausibility. We've shown all of that. Now let's think about clinical usefulness. Is there something we can do? Absolutely, yes, we can. If we know that our phase 2 detoxification is impacted, we can look and see what can we do to upregulate phase 2. Because even though GSTM1 might be deleted, there is a whole family of GSTs. And if we can upregulate or switch on the other GSTs to work harder and better, using things like sulforaphane and our cruciferous vegetables, we can compensate. So we knew something, and now we make a very clever decision with our patients. Right. So in my opinion, GSTM1 gets both a tick for scientific validity and a tick for clinical utility. Now let's talk about this gene. The little bit controversial. This is the LCT gene or the lactase gene. So I'm pretty sure for those of you who have been working with genetic tests up until now, that every single genetic test that you have worked with has what they call the lactose gene in, or the LCT gene. And the reason being is that it is believed that the lactase gene, so the lactase gene is evolutionary. So I'm not sure if you know this amazing story, because genetics is really just about evolutionary biology, that up until when our ancestors were hunter-gatherers, we were not able to break down lactose. We did not have any dairy in our diets, and we could not break down lactose. And when they moved to Northern Europe and started settling down and domesticating animals, there was an evolutionary advantage to be able to milk those animals and make products out of the milk and increase their nutrition by using those animals' milk, which obviously was an evolutionary advantage. And what we saw was this sporadic mutation, this change, this gene variant that happened, that suddenly we were able to break down lactose and be able to consume dairy, cow dairy. Now, the issue is that didn't happen for everyone. And we know that there are certain people who are what we call lactose intolerant, others who are lactose tolerant. They call it lactose non-persistence, which is, did the lactose enzyme persist or did it not? So because it has a genetic variation, it makes sense that this is the kind of thing you would want to see in a genetic report. So do I have the gene variant that tells me whether I can break down lactose or not? And that's why it's always been reported in genetic tests. So scientific validity, absolutely yes, get a very big tick. There is loads of research showing that this gene variant changes the enzyme activity, the lactase enzyme activity. But here's the issue. Now let's talk about clinical utility. So about 5 years ago, I set up a clinic in Cape Town called CTG, Center for Translational Genomics. And the idea of the clinic was to see whether what we were teaching in our nutrigenomic courses actually worked in clinical practice. So it was myself, it was 3 dietitians, and a functional medicine doc. And we were using a genetic test from one of the previous companies that I'd built tests for. And what we started noticing is that even though the genetic test would say that the person is likely to be lactose intolerant, when we asked them about it, they were absolutely fine with drinking dairy. So we started paying more attention to this and saying, well, why are we seeing such a lack of correlation between those who shouldn't be able to tolerate dairy and those that can? So when we built Darwin, the first thing I did was go and look at the Darwin scores for the LCT gene. And when we looked closely at the science, we found that even though it's true that the LCT gene variant will impact the enzyme activity, how it impacted the enzyme activity had a huge variability. So the same genotype, the same genetic result could result in almost no lactose, a lactase, so a real very small amount of lactose, so they were not able to tolerate dairy. But with the same genotype, they might have a much higher amount of lactase and be able to digest dairy well enough to be able to be functional. So in this case, we couldn't correlate the genotype, the gene result, with how much lactase it was, and therefore we couldn't trust it. We remember we needed to have something that was clinically useful. We needed to know that if we gave the recommendation, the likelihood was that the phenotype would be affected. So for that reason, the LCT gene got a fail, and in the 3x4 genetic test, we do not have the LCT gene. This is something else that we did. We made sure that we build a very, very tough scientific advisory boards. People like Jeff Bland, Helen Messier, Miles Spar, Ruth DeBus, Sarah Gottfried, Kathy Swift, Christine Horton, that holds us accountable. So every couple of months, when we present to our scientific advisory board, we need to be held accountable by what we're building, what genes we're including, how we're communicating it, where we're testing, and what we're testing. So this was just another layer to ensure that we would make sure we had science that you can trust. So the second part now is clinical translation. So now that we've got science that we feel is foundational and robust and rigor, how do we communicate it to your patient? And one of the problems up until then was that every genetic test had like a PDF with a table, a gene, either 3 stars or 3 droplets or traffic light, and it looked like every other test that you get from Quest or LabCorp. So we wanted to create a way to communicate with patients and practitioners that would allow for a different engagement with genetics. And we did this in 2 ways. The one is by using the language of color. So you'll see everything in the report is color coded. Now you've got, I believe you've got access to the sample report there, and you could download it and open it, and you'll see that everything we have done has got a color code. And we talk about color and impact. So the dark purple will have a very high impact, not risk, impact. And light purple, high, blue would be a medium impact, and yellow or green would be a low impact. And this gives you, the practitioner, a way to have a conversation with your patient that is using a language that is very, very easy for them. It's not using gene variants and enzymes and translation and transcription. It's just using the language of color. And the other thing we did was we wanted to use infographics, because in our clinic, we used to draw pictures all the time. And we realized that if we're drawing pictures all the time, why don't we simply create images, which is what we did. So we wanted to create an easy to understand report, both for you, the practitioner, but also for you to be able to explain where to start. So we have, I'm actually going to flip forward, 36 pathways. Remember, we built a pathway-based approach, and you'll see here 6 categories. Remember, I said we are a functional nutrition, functional medicine-based approach. We always go upstream. We go start at the top. So for us, starting at the top is cellular. What is happening at a cellular level? Then we go down to the next level. You can see it down the side. You can see the names. A systems approach. What are the systems we want to approach? Cardiovascular health systems. Then we look at the energy pathways. Some people might call this weight or diet, but we like to think of energy. How do we consume energy? How do we burn up energy? We have a category of activity, and then we have a category of nutrients. For every single pathway that you're seeing here, it is a polygenic risk score. There is a group of genes that have been scored to determine what impact color your patient is going to have. And then we identify what are the 3 most impacted. Now, you as a practitioner are going to take these pathways and still know who your patient is. So you don't use genetics in isolation. Genetics is not an answer in itself, but it is a guide even for a conversation with your patients of where to focus, where to start. And for those of you who don't know why we called 3x4, the idea is that's 3, 3, 3, 3. What are the 3 most important pathways I want to start with my patient? So when we look at this and we see the very high impact, the dark purple, and we want to start upstream, I may decide to start with detoxification, inflammation, and depending on the context of my patient, I might choose hormone balance. So the first 3 is what are the 3 pathways to start? The second 3 are what are the 3 most important diet recommendations I can make? The third 3, what are the 3 lifestyle recommendations I can make? And the final one is what are the 3 supplements I can recommend? These are just guidelines, but what we're trying to do is take a complex science and make it simple by capturing it in really a one-pager. And then the other thing we did was we wanted to capture the conversation, and we did that by creating these infographics. So for every category, there is an infographic. So in this image, you can see cellular and energy. Every single pathway has its own image. And the really important thing to understand here is that every infographic is different for every patient, because depending on the genetic results for your patient, their picture, their image will look different. And that's part of the excitement, is even for patients to compare their own reports and say, does your picture look like this, or why does your picture different? And we teach a lot about, and I will talk a little bit about where you can go to learn these things. What are the pathways? What are the images? What does it mean when it's looking like that? This is a great conversation starter for your patients when you're trying to talk to them about the importance of the pathway and the changes you would like them to make. This is our activity pathway. Everything is based on the concept of storytelling. There is a story to every picture that enables you to communicate it with your patients. And this is our nutrient one. You have the sample report and you'll be able to go in and have a much closer look. So ultimately, there are two parts to genetics. The first is insight. We do a genetic test to look at the gene variants to understand how we differ from each other, to understand how we respond to the world around us. And by that I mean, how do we respond to the food we eat, the toxins in our environment, the exercise we do, the stress and the trauma that we may endure. We understand more about our patient, and our patients understand more about themselves. And the second part is about taking action. What are we going to recommend for our patients that is going to upregulate their genes, switch on and switch off genes to be able to really optimize their health? Ultimately, if what we are offering our patient does not allow them to know what to eat for breakfast, then once again, we are selling data and not clinical value. So at the end of this, and you will see in the report, there are diet recommendations, lifestyle recommendations, and supplement recommendations. Okay. So let's move on to the last few pillars. The education, which was education, mentorship, and community. So in education, we want to provide multiple ways for you to learn about nutrigenomics. So we start off with what we call our blueprint certification. This is only 90 minutes and basically gives you the basic principles of nutrigenomics, penetrance, nutrigenetics versus nutrigenomics, all the gene variation, all the basics you need to know. But then it also takes you into understanding how the report will build, polygenic risk scores, pathway-based analysis, upstream, how to interpret the different pages of the report, what the different pages of the report mean. So we call this the 3x4 blueprint. At Avexia, you are not required to do this 90-minute in order to use it in your practice, but we highly, highly recommend it. It gives you everything you need to know about using the report in your practice that will be difficult to get to without doing the 90-minute. And I believe there will be a link that will be available to you, that'll be sent out to you, which can literally, it's free, it's easy, it's self-paced. You can start and stop in any time. This is just some of the images from our blueprint. And then we have the 3x4 foundation. So for those of you who maybe have already put your toe in the water of nutrigenomics, you're really excited about it, but you want to gain more mastery, you want to have a deeper knowledge of nutrigenomics, we have what we call the 3x4 Ed Foundations in Nutrigenomics. It takes about 2 to 3 months part-time. It's self-paced. I think it's 11 modules, but really it'll give you greater depth of knowledge and mastery around the concept of nutrigenetics, which is gene variants, the SNPs, and nutrigenomics as well. How do you use nutrition to change gene expression? There's multiple other touch points that we do. We have many, many webinars. We're launching probably in the next week a podcast around genetics and functional medicine called The Power of Genetics. We have a fantastic ebook called A Practitioner's Guide to Genetic Testing. It's available for a free download off our website that you're all welcome to. Talks about some of the challenges around nutrigenomics, some of the opportunities, how to leverage genetics to build your practice. So you're welcome to go along and get that. And then we have multiple events. In addition, we don't just offer education, we offer mentorship as well. Because just learning the theory is not enough. When you start working with your patients, you're needing a way to be able to make sure that you've got a little bit of hand holding before you have the confidence to kind of go off and become expert. We have two extraordinary clinicians heading up our mentorship, Annalise Smith and Nikki Grant, both dietitians, and they have been working with Dr. Sedano to make sure that Dr. Sedano is trained up to the highest level possible in the 3x4 genetic report and how it works, and having access to all our materials. So they have been working with them for a couple of months now to make sure that Dr. Sedano is as well versed in 3x4 genetics as you are. So we also, in addition, offer other platforms for mentorship. So you are able to send through any questions you have that have come up with your patients, or just questions you may have. And we have every single week something called the Power Hour, where all the questions we get, we answer in an hour, either Annalie or Nikki, and we deal with anything that you send through, and we always have case studies built into it. So it is probably the easiest way for you to upskill and learn, and it is always recorded. And then we have something called the Gene Team, which is also run by Annalie and Nikki. And the Gene Team is about deep dive clinical. So every Gene Team is only once a month, but we deep dive into a single topic and how it relates to genetic testing. So our last one we did was on histamine overload. We've done a Gene Team on thyroid. We've done on hormones. And these are all available where they're always super practical, very, very clinical. They've always got case studies, and we always have experts come and join us. And then lastly, we have our 3x4 community. This is a Facebook page, which we would welcome you to come and join us, irrespective of whether you decide to sell the test or not. Please come and join our community. You will be notified of all our events and all our webinars. There's a huge amount of content. Every week we put a summary of the best article we found, and we summarize it for you. There's a lot of outstanding practitioners who are already on the community group that are there to answer questions for you as well. So you just need to go to 3x4 Community on Facebook. And if you are a practitioner, you will immediately gain access. And you will also be able to access Power Hour and Gene Team from Facebook on Facebook Live. So I hope that I will see all of you on the other side. I'm really hoping that you will join as we're getting on the action, that I will see you in our webinars, that I will see you in the community, at 3x4 community and Facebook, that I will hear from Dr. Sedano that you're engaging. And I really look forward. This is a journey, no matter if you're just dipping in your toe for the first time or whether you are further down the road and quite familiar with genetics. Just two things to bring to your attention. I have mentioned the 90-minute certification course quite a bit. Self-based, it's theory with some real-life case studies, and really helps you understand what the product is. We'll send you a link so you'll be able to sign up for that. And the other one is that we have put an introductory discount onto the first 3x4 genetic test that you sell from the Avexia portal. Our price point, our retail price point is $289, but for the first order that you place, it will be $249, and this will be done automatically. And please feel free to be able to use it for yourself, friends and family, or of course for one of your patients. And just to reiterate that, while we highly, highly recommend the Blueprint certification course, if you want to be able to really comfortably use 3x4, it is not a prerequisite for selling the test via Avexia. You will be obviously choosing the 3x4 genetic test via the Avexia platform. And I think, Joshua, that wraps up me. And this slide is entirely yours.
Well, excellent. Thank you ever so much for that incredible information, Dr. Joffe. That was truly exceptional. Before we actually get into the how to order and the process, let's take a minute to address some questions, because you really packed a lot of information. It's extremely helpful. I know that there was a few questions that came in, and I know that Dr. Sedano is also on the call with us today, so I think he might have some things that he'd like to share as well, so we'll give him a chance to chime in at the moment. So the first question that I see that is clinically pertinent is, if a patient is compliant with the recommendations, would the report look different in 6 months to a year if they were to redo the test?
It's a very good question. So the great news about genetics is you don't redo the test. So this is one of the differences between genetics and functional testing, is your DNA does not change. So if I did the test at 6 months old versus 60 years old, it would be exactly the same outcome. So this is a once-off test for your patient. You will use, which actually raises something else for me, which maybe Dr. Zadana can speak to, but you will use other functional tests or other testing, basic biochemistry, to see whether the recommendations that have been inspired by the genetics are working or not. And that brings me to something that I did forget to tell you, which is that there will be a document that links the 3x4 genetic tests with all the tests that are being offered by Avexia. So we talk about almost using genetics as a screening tool to identify, what don't I know? What don't I know about my patient? What are the areas that I should focus on? Where should I start working with my patient? And then potentially, what other tests should I do with my patient that would give me more insight into whether my intervention's working, whether that really is being impacted. And this is a document that Dr. Sedano and Annalie and Nikki, our mentors, have been working on, that will create the link between all the other tests that are available on the Avexia portal and the genetic test. But the genetic test only needs to be done once.
Fantastic. All right, the next question. Do you look at both male and female sex hormones in all patients? For instance, male hormonal genes in women, or genes impacting estrogens in men? And if not, why?
So we actually have separate reports. We have a male report and we have a female report. And we have a hormone pathway in the report. And originally we actually thought we'd have different genes, but the reality is that both men and women have all the different hormones, but how they impact the different male genders is different. So the recommendations and the translation and the impact will be different, but the genes are actually the same. It's just the scoring, right? The Darwin scoring will be different, and the impact and the recommendations are different depending on whether it's male or female. But we absolutely do have 2 different reports for male and female, specifically because of the question you raised. So you were absolutely spot on.
All right, let's move on to another question from the attendees. Dr. Joffe, one of the things that you touched on, is the pathways, but when you're looking at SNPs, can you tell us about how many SNPs that you're looking at with this test, and how that relates to the genes that you're looking at?
Absolutely. We have 134 genetic results, and not 134 SNPs, because some of the genetic results will have multiple SNPs. So we have something called a haplotype, which is where you group some SNPs together and it gives you a result, like in iron overload, HFE, or even ApoE. So we can say there are 134 genetic results that you will get in the report. And in the report, if you have downloaded and you have a look, if you go to the back of the report, I guess you could call the appendix, you will see a page called Genes by Pathway. And what you will see there is for all 36 pathways, you will see we have listed what the genes are that contribute to that polygenic risk score for every pathway. And that will give you an idea of which genes are contributing to the estrogen pathway, detox, methylation, whatever that pathway is. You'll be able to see which genes are being scored, and you'll also see that they are listed in their color. So say in the detox pathway, if you're looking at that, you'll see genes, and whatever obviously is in purple is having the greatest impact on detox. Whatever dark purple has the greatest, light purple, etc., etc. So we carry that color coding throughout. and you will be able to see what genes contribute to which pathway. And of course, all the 134 genetic results appear in the results closer to the front of the report as well.
Dr. Sedano here. I've been working with this for several months, and I have to tell you that I was so impressed by the work that was done and put into this. I have seen other genetic tests throughout the years, the last 15 years or so, and they were definitely difficult to work with and sometimes not clinically significant. And so I kind of went the wayside, and when this thing came along, when I was introduced to it, certainly was excited. And from a technical perspective, here's how I've used this. I've already used this test on a patient. So here's a sample, and this may help you all to, like, how do you test this out? How do you monitor this? Well, I want to use methylation as an example. Right? If there's a problem with methylation, and Dr. Joffe talked about methylation being important, the COM gene for detoxification, estrogen metabolism, also for the catecholamines and neurotransmitters, dopamine, epinephrine, adrenaline, so forth. Right? If there is a SNP in there, say methylation SNP, what would you do? Well, in my perspective, what I did was went ahead and ordered a test looking at methylation specifically, seeing is a patient replete? Do they have enough methionine? Is there a need for B12, B6, betaine? Right? So you can initially get an idea and do that test. Go ahead and treat, see how they're doing, and then monitor the outcomes of your treatment. Say 6 weeks, maybe 8 months. Maybe 8 weeks down the line. That can be done for a lot of things. Organic acid tests. There's a lot of tests that you can use based on this genetic testing to see how your patient is doing. So hopefully that's helpful to y'all.
Excellent. Thank you, Dr. Sedano. Okay, we're getting a little bit closer on time. I know that we had this scheduled till 4:30, but I think that we're about wrapping up here. Dr. Joffe, any further points that you wanted to make before we move on to how to order? Okay. All right, Dr. Joffe, if you would mind just forwarding the slide to the how to order.
Oh, sorry.
Oh,
I was actually on mute. I was actually saying something. So I just wanted to, if you don't mind, Josh, just one last comment.
please.
I just want to say that the way we built the report, you don't need to be an expert practitioner in nutrigenomics to be able to use it. We built it in such a way that you could be new to nutrigenomics and still use it. And as you gain knowledge and expertise, you will just use more of it. You will dig deeper. You will find more detail. But it can be used in such a way that even if you're new and you only use the pathway page. So what I always recommend is just jump in and do one patient. Dr. Sedano is there to help you. You have an amazing asset at Avexia to be able to support you. And we have this extraordinary community that can also support you in terms of continuous learning and mentorship. So I hope you'll just take that first step, no matter how slow it is, and come and join us.
Marvelous. Dr. Joffe, we actually had a few more questions stream in at the end. I hope you don't mind. I figure we've got a few more minutes if that's all right. Super. So one other question that came in, and again, folks, if you're still out there and you want the question, you have Dr. Joffe at your disposal right now. I would take advantage of this. So one question that came in was, are you using a higher therapeutic dose of supplements suggested because maybe the SNP is creating a problem with assimilation?
Well, that's quite a broad question, because obviously we would need to speak exactly what the SNP was. And remember, we try not to supplement SNPs. So we look at what pathways have been impacted. We look at what's happening with your patient in general, and then we'll make supplement recommendations. We're not big fans of high dose supplements in any way, especially not in the methylation space. So the most important thing about our test is we do not make recommendations for supplements based on single snips. We do make supplement recommendations. We don't usually give quantities because we feel you're the practitioner. We'll give a guideline of the kinds of supplements we would recommend, but ultimately you're the practitioner who's qualified to be able to work with your patient, and using ours just as a guide will make the final decision on what the best supplement is.
Excellent. All right, Dr. Joffe, another question that came in was, do you make a comprehensive client intake form and apply that to the results? Do you take into consideration medications and detoxification pathways, specifically thinking of aromatase inhibitors?
So another good question. So no, we don't. And the reason we don't is because we work with practitioners like you. So in a direct-to-consumer environment where there's no practitioner, you would probably want to try find out about the patient and what they're taking and what conditions they've got, and then try put that kind of contextual information. But the reason we're so committed to working with practitioners of the caliber of yourselves is because we know you're doing that. We know that you're doing assessments on your patients, you're finding out their medical history, you're finding out their diet history, you're finding out what supplements and medication they're taking. The thing around genetics is that, I can't say it enough, is genetics by itself does not determine your recommendations. Genetics is an incredible insight into your patient. It is knowing what you don't know. It is understanding how your patient may respond to the environment, but it is not an answer in itself. So you will add genetics into your assessment process for your patient, giving you a whole layer of insights that you didn't have, but ultimately you will be doing everything that you normally do with your patient, just adding in genetics as a way to understand your patient at a deep molecular level.
Excellent. Okay, now another question came in was, what is the sample type? So this is a buccal swab, if I'm not mistaken.
Cheek swab, that's right. So a buccal swab or a cheek swab, it's just 30 seconds on one swab that you rub 30 seconds inside the cheek. On one side, we really want to get cheek cells, and we extract the DNA and cheek cells, and it just goes into a little tube, and that's what goes off to our lab.
Perfect. Yeah, okay. Let's see, there was another question, and this goes back to something you had touched on briefly, Dr. Joffe, and I'm going to ask Dr. Sedano to also just, add his thoughts if he's able to come back online with us. But you had mentioned that there would be a resource, recommendations given for other available testing through Avexia, like say, for instance, an organic acid test or something of that nature, or anything else that we might offer that could help, with patient outcomes based upon the genetic results. Can you speak to that a little bit more?
Yeah, so I think Dr. Sedano probably can speak to, because he's the one who's working on the document, but the document's in process, and essentially what it'll do is for every pathway that you saw in our 3x4 genetic tests, there will be recommendations for which of the Avexia functional tests would be recommended. And the idea here is that you're almost using the genetic test as a screening tool. So where you're seeing like a very high impact dark purple, you may want to go and order additional tests to dive deeper. And that's where the Avexia will come in. So, and that's where the document. So I know that we're about halfway through completing the document, but perhaps Dr. Sedano can tell you more. He's a lot closer to the actual document than I am.
Yeah, sure. Let's go back to the methylation example again. So if there's a SNP in methylation, one way or another, again, it's a good idea. A basic test would show up, and that is called methylmalonic acid, MMA. That shows a functional need for folate, for methylation in the body. That test can be used and to see what are we getting back? What's the range, high or low range? Are we getting adequate functional, methylation in the body? That's how you would use that. And there are many examples using organic acid testing, even stool analysis testing, by the way, which is a whole nother. We don't have time for that right now. But yeah, that's how this is used. And we're pairing up the functional test really to monitor, to say how much supplementation would be recommended for this particular person, because we're talking about personalized medicine. You know, I'm sure you're familiar with biochemical individuality. You've been exposed to it, and that's what we're talking about. The functional test will help hone in the biochemical need for that individual based on the SNPs. And, you know, I love this term, this polygenetic score, and thank you so much for talking about it, because that's really what we're talking about here.
Fantastic. Thank you, Dr. Stano. Thank you, Dr. Joffe. Before I move on to the how to order section, I just want to let you know that there have been a number of comments that have come in from the attendees today, Dr. Joffe, that applaud you for your work and all of your effort, and I want to join in on that as well. Thank you so much. It's really astounding at what you've been able to do, and I can't even fathom the reach that this would have impacting patients and producing better outcomes. So thank you. Now that we've learned a little bit more about the how excellent this test is, I want to make sure that everyone is understanding how they can go in and order the test before we conclude. So as you can see on this slide, first you would need to log into your Avexia account. You would go to Avexia link, which is located under Quick Links. That's on the left-hand side of the dashboard when you get logged in. Next, you would select or create a patient to assign the test to. Then you would select specialty labs and find the 3x4 blueprint. And then finally, you would complete the test order by clicking the green Place Order button. Now, please keep in mind that due to the personalized nature of this test, all orders must be processed through a Avexia link and assigned to a specific patient. Now, also, as Dr. Joffe had mentioned earlier in the presentation, we are providing an introductory price for the first test that's performed under your account. You will automatically receive an account credit of $40 to reduce the cost from the regular price of the $289 to the $249. That happens automatically. So with that being said, Dr. Joffe, if you want to advance to that next slide, I want to make sure everyone has an opportunity to take a look at the many forms that you can utilize to contact us. We are always happy to assist you with any issues or questions that you may have, and we can be reached during our business hours between 8 a.m. and 8 p.m. Eastern Time at 888-852-2723, Monday through Friday. You can also contact us anytime via email at info@avexiadiagnostics.com. Thank you again, everyone, for joining us for this webinar event. Until next time, from everyone at Avexia Diagnostics and 3x4, stay healthy, stay safe, and we wish you all the best on your pathway to wellness.